I'm Dr. Nancy Davidson. I want to welcome you to ASCO's plenary session. As the immediate past president of ASCO, I'm also the chair of the 2009 Special Awards Committee. I have the honor, therefore, to open our plenary session with this presentation of the Science of Oncology Award and Lecture. ASCO's Science of Oncology Award and Lecture was created in 2005. It honors the contributions of innovative basic scientists in cancer research. This year, we are proud to recognize Dr. Bert Vogelstein with this award for his pioneering studies on the pathogenesis of human cancer. Dr. Vogelstein was the first scientist to elucidate the molecular basis of a common human cancer. In particular, he and his colleagues have demonstrated that colorectal cancers result from the gradual accumulation of genetic alterations in specific oncogenes and tumor suppressor genes. His group's discovery and analysis of these genes and their functions represents a landmark in the application of molecular biology to the study of human disease. Dr. Vogelstein is the director of the Ludwig Center at Johns Hopkins, an investigator of the Howard Hughes Medical Institute, and Clayton Professor of Oncology and Pathology at the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins. He's won numerous awards over the last several years for his outstanding contributions to cancer research, including ASCO's David A. Karnofsky Memorial Award. Please join me in welcoming and honoring Bert Vogelstein, whose award lecture today is entitled Cancer Genomes and Their Implications for Understanding and Managing Cancer. Dr. Vogelstein.
Oh, don't take down the teleprompter. I was really looking forward. Makes me feel important. It's an honor to be here and to win this award, of course. Appreciate Nancy's kind comments and her introduction. It's also a privilege to be able to speak to this audience. This is my favorite audience to speak to, for a very simple reason. As a basic researcher, the only thing we do that's important are those things that matter to this audience in the end. If they don't matter eventually, then they're really not that important. So what I'd like to do today is give you an update on the cancer genome, the data that's available, and hopefully stimulate you to think what might be done with this kind of knowledge in the future. And I'll divide my talk into three parts. First will be the data about the cancer genome as we now see it. These data have improved quite a bit over the last few years, because now all of the genes, not just a selected subset, have been subjected to detailed analysis, including sequencing. Second, how to interpret these data and put it into a context that makes sense. And third, how we might exploit this knowledge to help patients in the future. The data that I will discuss comes from analysis of 68 tumors, solid tumors, the breast, colon, pancreas, or brain. The data was mainly assembled by 6 talented trainees, 3 postdoctoral fellows, Tobias Schoblum, Sean Jones, and Will Parsons, together with graduate students Lauren, Jimmy, and Susan. And these trainees were supervised, they might say bothered by, a whole group of faculty investigators like me, most from Johns Hopkins, but through important collaborations with Sandy Markowitz at Case Western Reserve, Darrell Bigner and Haiyan at Duke, Phil Buchholz at University of South Carolina, and others. The data comes from 3 types of analyses. First, in-depth sequencing, second, copy number analysis, and third, gene expression analysis. The sequencing goes like this. There are about 21,000 genes in the human genome. The hardest part of this kind of project is designing primers that amplify each one specifically, that is, each exon or portion of an exon, requires a little more than 400,000 primers. These are used to amplify and sequence by conventional Sanger sequencing. And when done, the sequences are analyzed. Any putative mutations are resequenced to confirm their integrity and also to make sure that they are not present in the patient's normal cells. So here we're looking specifically for mutations that occur after birth in the tumors, but not in the normal cells, somatic mutations. This is done on anywhere between 11 and 24 tumors. We call that the discovery screen, and that's followed by a validation screen in which we study an additional 24 to several hundred cases depending on the mutation of interest. And we can now ask for the first time a relatively straightforward question. How many genes are altered in a typical solid tumor? And the answer for pancreatic cancers is 44. And I think you can see from this graph that there's a reasonably uniform distribution. There are some outliers to be sure, but I think more uniformity than one might have expected. Now, these mutations are subtle. They're single base pair deletions or insertions, couple base pair insertions or deletions, mainly missense mutations that change C to T, for example. There are other kinds of alterations, as you know. One kind is amplifications, and these are now indicated in green on top of the blue bars, and they're roughly 8 or 10 in a typical pancreatic cancer. And deletions, that is homozygous deletions, which remove both copies of the allele, are also present throughout the cancer genome. There are another 5 or 10 per case. Altogether, there are about 63 alterations on average in a typical pancreatic cancer, and the number in colon cancers, breast cancers, and brain tumors are broadly similar, somewhere between a total of 50 and 100 per tumor type on average. Now, I have a little bit of trouble visualizing charts like this and trying to actually see them in a context that I can recognize. So let me share with you how I visualize them. Perhaps it will help you to do the same. I look at the human genome as simply a series of books, an encyclopedia, where each page, each gene is a page containing about 1,500 A, C's, T's, or G's, and each book is 1,000 pages, which means it contains 1,000 genes. And there are, of course, 46 books, 23 from the mother and 23 from the father. And if you look at the cancer genome as an encyclopedia, it might look like this. Of these 20,000 pages, actually 40,000 if you count both the mother and the father's alleles, there are only 44 with typographical errors, these subtle changes, 11 missing pages, and 8 duplicated pages. That's really not very much. If you were to make an encyclopedia and only had this many typos in it, you'd be pretty satisfied. On the other hand, it means that almost all of the genes in a cancer cell, much greater than 99.9%, are the same as in normal cells. It explains, in part, why cancers are so much harder to treat than bacterial infection. This is a graphical representation of the same thing, where the blue dots represent point mutations, and the green dots represent deletions, and the red dots represent amplifications. I'll get back to this in a second. The complexity comes not from this encyclopedia, because this cancer genome represents only one patient. This is one particular patient who had 70 mutations in his tumor. The problem and complexity comes from the fact of the heterogeneity in the genetic alterations per tumor, which I think you can see from this slide. Some patients have 100 mutations, some patients have 40. But the main complicating feature is that if two patients both have 65 mutations in their tumor, only 3 or 4 of those mutated genes are common. The rest are different. A great heterogeneity. The couple hundred genes that can contribute to solid tumors are mutated in individuals in every possible way that you can think of, making the genetic makeup of each tumor unique and undoubtedly responsible for some of the heterogeneity that all clinicians are well aware of. No two cancer patients are the same. So one of the major questions that we face as cancer geneticists is how we can interpret this and, again, view it in a way that makes sense. And there are two simplifying features. First, only a fraction, a relatively small fraction, say 20%, are drivers. By drivers, I mean mutations that occur and cause the cell to be born more than it should be, that is, increased cell birth, or die less than it should, increasing net cell growth, increasing a selective advantage in formal evolutionary terms. The other mutations are simply passengers. These are mutations that occur just as cells divide. Cancer cells, just like normal cells, whenever they divide, they make mistakes. Most of those mistakes are neutral. They cause no increased fitness or decreased fitness, but they're along for the ride if they happen to occur in a cell that has a mutation that does confer a selective advantage. So they're passengers. And if we look at the cancer genome in that way and subtract out all the passengers, it looks like that. There are only about 12 or 15 mutations that are left that are drivers. And trying to figure out which are the drivers is not an easy task, but gradually techniques for doing that are emerging. So we're now left with a dozen or so mutations in the cancer genome that are presumptive drivers. There's another simplifying way to look at it, and that is to look past individual genes and focus on pathways. Now, pathways, as you know, means the mechanisms underlying the gene's action, either upstream of the gene or downstream of the gene. And this illustrates the PIK3CA pathway, a gene that is itself mutated in many cancers, but along its pathway, that is upstream receptors that control its activity, other mediators, downstream effectors, many of them are also mutated. And they're generally mutated in a mutually exclusive fashion. That is, if you have a PIK3CA mutation in a cancer, you don't have an mTOR mutation in the same cancer, etc. And in pancreatic cancers, actually, as well as the other solid tumor types we studied, there appear to be a core set of pathways which are quite similar from tumor to tumor, although the details vary. So in pancreatic cancers, one can define 12 core pathways. which are commonly mutated. That is, mutated, at least one gene from each pathway is mutated in most pancreatic cancers. Here's an example of the TGF-beta pathway, which is mutated in 100%, as far as we can tell, of the cancers that we've studied, the pancreatic cancers. If you look at individual tumors, though, you see how that 100% is made up. Here's one tumor called PA14 and another tumor called PA10. PA14 has a mutation in SMAD4, which is a transcription factor member of the pathway, whereas PA10 has a mutation in a receptor that's right at the top of this pathway, BMP receptor. And in that manner, each of these pathways was found to be mutated in the majority of pancreatic cancers that were studied. Similarly, one can compose pathways, many of them identical to those that are altered in pancreatic cancers, for the other solid tumors. And this brings us quite naturally to the third and most important part of this talk, which is ways to exploit this knowledge. The first is with pathway-oriented therapeutics. These core pathways are all there is, apparently. And the reason I say that is because our group, as well as other groups doing large-scale sequencing studies, such as the TCGA in the United States and the Sanger Initiative in the UK, have discovered many new genes over the past few years. But none of us have discovered any new pathways. Every time a new gene is discovered to be mutated in cancers, it's only a very short period of time before one is able to put that gene into one of these relatively small number of pathways. What that means is that the pathways are saturated in a genetic sense. We know them all. There isn't any more. This is what cancer is. That is kind of sobering for many reasons. The practical implication for therapeutics development is perhaps the idea of using more pathway-type gene drug discovery rather than individual gene targeting. Most pharmaceutical companies, as you know, are targeting individual genes, kinases and the like, for very good reasons. But the advantages of targeting pathways with cell-based screens rather than with molecular screens lies mainly in the fact that individual pathways are going to be altered mainly by activation in most forms of a cancer, whereas individual genes are going to be suitable for targeting only in a much smaller fraction of any individual cancer type. So I think in the future we're likely to see more targeting based on pathways rather than individual genes, or at least I hope we will. Another way to use this information is through immunotherapy. Jim Allison did a study last year looking at the mutations that we identified in colon cancers and breast cancers and trying to find which one of the missense mutations would likely be represented by the immune system, and found that roughly 5 to 10% of these missense mutations would be predicted to bind to any common HLA epitope. The idea behind this is that, say, in a few years, when cancers become much easier to sequence, one can determine the sequence of all the genes in a cancer, find 5 or 10 genes that have missense mutations that should be well presented to the immune system, and make a vaccine based on that particular that particular tumor's mutated gene and its ability to be recognized by the immune system. That sounds futuristic, but the advantage of it is that these mutations are found only in the patient's tumor cells, not in any normal cells. So if an immune response could be elicited to such a peptide, then it should be exquisitely specific for the tumor. For those of you who are interested in such things in the future, I can tell you how much it costs right now to sequence an individual cancer. It costs about $100,000 and takes about 2 weeks. In the next 2 years, that cost will come down by at least 5-fold, thanks to next-generation sequencing. And in the next 10 years, it is likely to come down quite a bit further. So some of these futuristic ideas are not as futuristic as they would have seemed just a couple of years ago. Another way to use cancer genomics in the future is through the identification of patients or individuals who are predisposed to get cancer, particularly predisposed at an early age. Now, these heritable alterations are only going to account for a relatively small fraction of the total cancers, that is, solid tumors, but they're a particularly important fraction because they often affect younger members of the population and because they are, at least in theory, preventable. And let me give you an example of how to apply whole genomics, so to speak, to a case like this. This study was done by Alison Klein at Johns Hopkins, working closely with Sean Jones. She noticed a patient who had pancreatic cancer in his 60s and had a sister who had pancreatic cancer in her 40s. There were no other pancreatic cancers in the family, and the family was much too small to do linkage analysis or standard kinds of genetic analysis to identify the causative gene. So Alison and Sean, instead, after eliminating the usual suspects, decided to sequence all of the genes in these patients. Now, before I was talking about sequencing the DNA of the cancer. Now I'm talking about sequencing the DNA in normal cells. And they sequenced all 21,000 genes in the normal cells of the proband, the male. And looking at it in this encyclopedic context, they only found 3 mutations that made sense based on comparing germline DNA of this patient to the same patient's tumor DNA. The only 3 that were likely or conceivably likely to play a role in tumor predisposition. And one of them was particularly provocative because it was PALB2. PALB2 had not previously been implicated in pancreatic cancers, but it had been shown by David Livingston's group at Dana-Farber a few years ago to encode a protein that bound to the breast cancer predisposition gene BRCA2. So this was an excellent candidate, and in fact, Alison and Sean found that both these patients had inherited a stop codon in this gene, that their tumors had deleted the other allele or inactivated it by a point mutation. And when they looked at 96 other small families who appear to have some sort of hereditary predisposition to pancreatic cancer, they found 3 more with identical features. That is, germline mutations, stop codons in one allele. These mutations were not found in more than 2,000 normal individuals. Many of you have heard discussions about the use of personal genomics almost at birth to identify the sequences of all genes in people as they're born to try to predict who is going to get what disease. That will certainly become possible within a few years. Whether it makes sense to do so is a much more difficult question. But in a family that already clearly has some possible predisposition to a disease, in this case pancreatic cancer, but you can think of many more, it might make sense to do that, and it certainly can be valuable to do that, as illustrated by this case. Other members of this family can now be screened for the same mutation, and those who are found to have it can be focused on in terms of surveillance, trying to detect the tumors early enough so that they can be cured. And this brings us to the last topic that I'd like to discuss today, which is early diagnosis. This is the one that is closest to my heart. I think that 20 or 30 years from now, cancer won't be the problem that it is today, not because we can so easily cure advanced metastatic cancers, but because cancers are detected earlier, at a stage when they can be treated with surgery, radiotherapy, and adjuvant therapy. And I'm optimistic about this approach because of the timing, of the long times it takes to develop metastatic cancers. This data is from a study done by Sandy Markowitz, published last year, on colorectal cancers. Colorectal cancers, as you know, have a well-defined series of genetic events, starting with mutations in the APC beta-catenin pathway, and following with mutations in other genes such as RAS and PIK3CA and P53, etc. Now, by using techniques borrowed from evolutionary biology, Sandy and his team could track the timing of these mutations, and found that it took approximately 30 years to accumulate the 60 or 70 mutations that were present in most of these cancers. They did this by looking at the same cancers at various stages of their development. The adenoma from which the cancer developed, the original primary tumor in the colon, the metastatic lesions in the liver and the lymph node, etc. And by knowing the mutation rate, you can relatively easily figure out how long it takes to get that many mutations. And it takes 30 years. This is consistent with many previous clinical studies, radiographic studies following patients with FAP, familial adenomatous polyposis, or patients with adenomas that were untreated. And all of these studies suggest a very long time from initiation, that first mutation in the APC pathway, to the advent of metastasis. Any patient that dies from colorectal cancer dies only because that patient's tumor was not detected in the first 27 years of its existence. It was only detected within the few years prior to its causing death of the patient. At the same time, that provides this huge window of opportunity, that 27 years, to try and identify a tumor at a stage before it metastasizes and when it is amenable to techniques that work even today. Again, surgery, radiotherapy, adjuvant therapies. And there are many ways one can imagine doing that in the future. The first is through molecular imaging. That is, trying to image tumors not with FDG PET, which lacks specificity for screening, but with tracers that are more specific for the activated pathways that occur only in cancer cells. And obviously efforts are ongoing to do that. Another way, which we have explored in our laboratory, is through the detection of the mutant genes themselves in various bodily compartments. And this effort has been led by Luis Diaz in our group, with several other faculty members and trainees contributing over the last few years. The main approach that we use is called digital genomics. We try to look at DNA molecules one at a time, rather than putting them all in a pot and looking at them together, which is the classical way of looking for mutations. We look at them one at a time. That's why they're called digital. We count them. And we do that by transforming the individual DNA molecules to beads and then look at the beads through conventional flow cytometry. And here's an example of a patient who was evaluated. The normal alleles are those red dots representing beads, which are stained with a hybridization probe for a normal allele. And the black dots are beads which are stained with a probe for mutant alleles. You can see that they're very well separated, and you can precisely count how many mutant molecules and how many normal molecules there are in any clinical sample. And here are some representative results that Louise has obtained in the plasma and the stool of patients with colorectal cancers. We're looking in plasma, of course, at the DNA molecules which are floating around in the bloodstream free of cells and that presumably have come from tumors. In advanced stage cancers, that is metastatic cancers, it's easy to detect these molecules. Virtually 100% of such patients can be detected through this simple blood test or with a test based on the DNA from stool. The problem, of course, is that this is not what we really want to do. The horse is already out of the barn when the patient reaches Dukes D stage. This technology could be useful for following patients with disease, as blood mutant DNA provides a very sensitive way to look for occult disease, but it's not useful for early detection. In early stage cancers, the blood test at present can detect about 50% of patients, whereas the stool test is somewhat better, about 90%. Now, 50% will hopefully improve, but I would submit to you that if one could institute a blood test for colorectal cancer as well as many other types of cancers for which no screening tests are available, then one could, at least in principle, save a lot of lives, even if imperfect. For adenomas, clinically significant adenomas that are premalignant, of course, the blood test is not very good. It seems to require invasion to get DNA molecules from a tumor into the bloodstream, but the stool test is pretty reasonable with about 70% sensitivity. Perhaps the most important part of these data is that there are no mutant molecules ever present in either the plasma or the stool of normal people. And that is, of course, because mutant molecules are completely specific for tumors. They're not simply markers for tumors like PSA, for example. They are causally involved in tumor formation. You can't get a tumor of the colon unless you have an APC pathway mutation. You can't get a tumor of the colon if you don't have a RAF pathway mutation, etc., for about 10 of these pathways. So they are, at least in principle, exquisitely specific and rational markers to use for these kinds of studies. So that's where we are. We've learned a lot about the cancer genome, but there's, of course, much more to be learned. Some of the challenges from my vantage point are distinguishing drivers from passengers, which was one of the most difficult tasks in cancer genomics, especially for low-frequency drivers, which are the predominant type observed in solid tumors. Second is this pathway definition. There are clearly only about a dozen pathways that are deranged in tumors, but we really don't know very much about how they work in human tumor cells. We often know a lot about how they work in Drosophila cells, or worm cells, or even mouse cells. But really, much less is known about how they work in human cells, and in particular, human tumor cells. One thing that has been learned by the great amount of signal transduction pathway research over the last decade is that there are only a small number of genes, as you know, only 20,000, and that their function in different cell types is not the same. It's not the same in different organisms, it's not the same in different cell types, and in fact, it can vary with time. I think one of the challenges for the future is understanding how these pathways work in detail in the actual cancer cells in which they operate, rather than in model systems. And the third challenge, of course, is translating this knowledge to patient benefit. This challenge is much harder than the other two because it requires much more creativity. The other two are driven, so to speak, by many previous studies of how genes could work, but there's much less known about how to translate any of these defects into real patients' gain, either through improved diagnosis or therapeutics. Some may consider these challenges. I like to look at them as opportunities. There's a great old story about an American businessman who was asked to sell shoes in Russia in the late 1800s, and his colleague told him that you can't sell shoes to Russians because most Russians don't wear shoes. And he said, that's exactly the point. This is an opportunity. And it's the same with this. Translation is an often used word, but it's not done as much as I think it should be or it could be, because it's so challenging, number one, and because it takes the expertise of many individuals working together. But I'm very optimistic, in fact more optimistic now than I've ever been about this possibility, because for the first time we are not blind men and women looking at different parts of the elephant. We can see the whole elephant. We can see all of the genes. We can see all of the pathways, at least in outline form. We know this cancer beast. Sure, there's a lot to learn, but one of the things that humans do best is take a little bit of knowledge and do something incredibly useful and wonderful with it. And my expectation in the next 10 to 20 years is that this new knowledge of the cancer genome will continue to be translated and transform the way patients are managed. Thank you for your attention.